to join with audio. Nope. Should be automatically. Start video. All right, y'all. Let me share the screen. Okay. And let me get iClicker going. We're almost there. Okay, show me what it is. Over here. This is labeled as alpha D glucose. Should it not be beta? Because it's up and up on the anomeric carbon. It's up and it should be, yes, it should be beta. But the book's wrong. The book's wrong. The book's wrong. It happens. The book can be wrong. Should I label it beta or just, like, is it supposed to be alpha in this case, or just change it to beta? It should be. Now I can erase this and put a
Right.
H if you want me to keep this label as alpha.
It honestly doesn't matter, because when you go from the straight chain to the ring, you could form either alpha or beta. As long as you're labeling it as D glucose and this one's D and this one's D, then it's fine. So you could do either way.
Okay,
Either way is correct.
Beta, so, right.
Yeah.
All right, how we doing today? Got my chat going. I get my, oh, turn the display on for everybody in class. That'd be nice. Stressed. Take a deep breath. You only have a little more to go. Just a little.
Yeah, no, it's covered glass.
Stay with it. Don't give up. Don't give up. We're close to the end. Please don't give up. Y'all got this. I know the stress level is high. I feel you. I really, really, really feel y'all. I do.
Sure you feel it?
I am feeling it. Trust me. Professors feel the stress just like y'all. And being a first semester, trust me, I feel it too. Okay, so, the important bit. Let's get the important bit done today. So why did the 2 4s sit down at the table but not have lunch? Because they already ate. That was, Charles laughed for like 20 minutes at that. Like, he just thought that was the best joke ever. I don't know. All right, you got to put your name, Zoom user. All right, let's see. So first things first is attendance. Let's see if I can hide this. Hide that. All right, and this, so many things. Okay, so go ahead and click in on attendance when you get a second, because we are going to need this because we're going to do some clicker stuff. So you should have your answers to your long clicker questions, right? And I have a few short clicker questions. They're easy, get us rolling, right? The ones on the bottom. Did I give them to y'all? Did I really?
I mean,
They were easy.
I was being able to just do clickers questions.
Dang it. Dang it. All right, we'll go over them at least. We'll go over them at least. I cannot believe I gave them to y'all. It happens. Okay. I need these. So they're supposed to be for me and not for y'all, but apparently I didn't do that so well. So let's do, it was like 30 something. Oh.
Did we ever do the traffic on like longer clickers?
That was one thing we were supposed to do in class, but we didn't have time to, so I posted the answers.
Wait, which was the answer?
In the announcement forum, I also sent you the link this morning.
Okay.
I did. I sent you the link this morning.
Okay.
Okay, so please, please, please go ahead on iClicker. And then we will
We'll go over the clicker questions. I'm not even going to bother doing clicker if y'all have already had them. Okay. All right. So y'all ready? Yes? All right. So since the citrate cycle is an anaerobic process, where does it occur?
Mitochondria.
Mitochondria is correct. What does anaerobic mean? Anaerobic. What does aerobic mean?
Means within the presence of oxygen.
With oxygen. Now, since you already have the answer to the iClicker question, let's talk about this a little bit. So why do we have to have oxygen present in order for this to work? Like,
Because the oxygen or the
it's our
oxidized
final, what? Electron acceptor, right? Because it has the highest, what?
the highest reduction.
The what? Reduction
Highest reduction.
potential, right? Because we're always going to go from electron carriers that have low reduction potential and then bump them up to higher and higher and higher, right? Okay, so give me an example of a place where the citrate cycle does not occur. So citrate cycle does not occur. So it has to occur in mitochondria. So you've given an organelle location, but red blood cells is correct because red blood cells are bags of hemoglobin. I mean, they don't have nuclei, they don't have mitochondria. They are poor, sad excuses for a cell, if you don't mind me saying. So
Hey, you carry oxygen throughout your body.
they do a great job for what they do, but they are a sad excuse for a cell. So what's another situation where you would stop going through citrate, where you normally go through citrate, but at some point you're going to stop? This one's a little more tricky.
Wait, where it stops?
Yeah, normally in the body, your cell's going to go through citrate cycle in this location, but under certain conditions, you'll stop going through citrate. No. Well, think about aerobic and anaerobic. If you're going to the gym, you're going to either, that's right, you're going to either go do your aerobic exercises, or maybe you're going to go do what? Anaerobic. So what situation would lead to anaerobic muscle? It would result in the buildup of lactate. Yes, it would. But like you would have to, your muscles are going to use oxygen, right? No, every time you exercise, you're going to use oxygen, right? And so that's aerobic. Now, what happens when you weight lift, when you stress your muscles so much that they run out of oxygen? Then you're going to switch to using glycolysis, right? You're going to switch over and you're going to stop the citrate cycle because you ran out of oxygen. So normally your muscle cells are going to go through the citrate cycle. But if you exercise so much that you deplete the oxygen, then your muscles can't go through the citrate cycle, but they still need ATP, right? That's where they'll get their ATP from. All right. Did I really give y'all all these? All right. One of those, see, I'm under stress too. I didn't notice. All right, high levels of succinate would result from a deficiency of what enzyme? So succinate dehydrogenase is the correct answer. What reaction is this? So tell me the reaction that is inhibited. So you go from succinate.
Is it dehydration reaction that where you absorb milk H2O, or is it hydration or is dehydration when you release H2O.
Well, think about what happens if you're gonna have a dehydrogenase. If you're gonna you're gonna take off a hydrogen from succinate. What's gonna happen?
Then you produce H2O. So if you take a hydrogen off.
Okay, wait. Let's do this, and then we're gonna answer that. Succinate goes to what? Fumarate via succinate dehydrogenase, right? So if you inhibit succinate dehydrogenase, you're going to stop making fumarate and you're going to end up with a pileup of succinate. So you could be asked that for any reaction, whether you're talking about the reactant or the product, okay? And you have the pathway, right? Everybody saw that I posted the 2 pathways. I'm giving you the names of every substrate and the names of every enzyme for your exam on Friday, not Thursday, Friday. Okay, yeah?
Yeah.
All right, okay. So now, Ross, your question was?
I was asking if this is a dehydration or a hydrate.
So what happens if we go back? Let's go back to that. What number is that? In our, let's see. What slide was that? That was 21. So we're going to come back to 34, 21. Okay. Nope. I'm off by 1. We'll go back. Reaction 4, reaction 5, reaction 6. Okay. So what's happening when we go from succinate to fumarate? It's not all about water. It's all about what?
Hydrogen.
Hydrogens. So here we have 2 hydrogens attached to each carbon. And when we undergo this reaction with succinate dehydrogenase, what happens?
You're moving on hydrogen.
Not just one.
From each, I was going to say you're moving hydrogen from each of the carbon.
Correct. And so then FAD is going to pick up both of those hydrogens.
Hence the double bond forming.
Correct. So it's not, just because you're moving hydrogens around does not mean that you're
using water.
Okay. All right. Okay, so what number did I say we need to go back to? 34? Yeah, all right. Okay. All right. Citric acid is found in high levels in fruit juices. How would drinking several glasses of orange juice affect the citrate cycle? Citric acid, depending on the pH of the solution, right, is also called what? Citrate. Citrate, right? It's whether you got the hydrogens attached or not. That's all it is. That's the only difference. So what would happen is you would pile up citrate. So the more citrate you have, if you have elevated levels of citrate, what's going to end up happening?
You're gonna feed into the cycle and
You're going to go into the rest of the Krebs cycle more quickly.
Is this part of the reason they say drink your orange juice? Is this why they always say drink your orange juice?
Orange juice has other good things in there for you, but it's got a lot of sugar in there too. So, you know,
Well, I mean,
eat your oranges. How about we eat our oranges, right? That's a good thing. All right, let's see. I'm sorry. And carbs. Yes, that's right. And carbs.
everything has carbs.
Well, you know, pretty close, right? All right. So what else is true? This one's true. C is true. Step 1 would be bypassed, right? Because you already have citrate. So you don't need citrate synthase to do its thing. So B and C. Why would the cycle go in reverse? That makes no sense, right? Yes, as long as that makes no sense to you. Woohoo. Okay. We did that. All right. Why does the citrate cycle produce more energy than glycolysis? How much energy do we get from glycolysis?
We get 4 ATP. There's 2 ATP.
We get a net of 2 ATP from glycolysis. We get more than that from the citrate cycle. We haven't gone into that yet, but we produce all kinds of energy carrying molecules, right?
Yeah.
And then where are these going to go?
They go there.
They are going to go to the electron transport chain. And we're going to make lots of ATP. That is going to be the name of the game. Make ATP. Yeah, it's a lot. It's good. All right, so
But they're both, aren't they both cyclic?
for starting with one glucose molecule, all the way down to electron transport chain, you'll get 32. So you get 30 versus 2. Not in the citrate cycle, but once you go through oxidative phosphorylation, that's like the end of the road, right? So the end of the road is basically how much ATP we make. Well, we know we go through from glucose through glycolysis, we're only going to make 2. But if you write the overall reaction, you're going to get 32. So 2 of those are from glycolysis. What? Yeah. Yeah, there. But we're gonna, I promise we're gonna go over it. It's the, yes, what's after is from the citrate. Starts in the citrate. We'll get there. All right, and what else is really great about the citrate cycle? It's cyclic, so you can keep going through it. So both A and B, so you can keep making those energy carrying molecules. B, what did, oh, yeah, thank you. I didn't scroll down. What helped to scroll down? Come on, there we go. Okay, all right. Okay, low levels of TPP. What is TPP? It's a coenzyme that is present in pyruvate dehydrogenase. Pyruvate dehydrogenase.
CPP is a necessary cofactor for pyruvate dehydrogenase.
Is it a necessary cofactor for the decarboxylase?
No.
No.
Yes, it is. The whole thing is. Yes, it is. Yes. Yes. So all of our cofactors are the B vitamins except for one. Yeah. The alpha lipoic acid. That's right. Okay. So how about acetyl-CoA levels?
Are important to decarboxylase except for the acid.
No. We're just saying that all of the cofactors that are involved are vitamins except for the alpha lipoic acid. Because we can make it. It's not a vitamin because we can make it. All right, acetyl-CoA levels would be reduced. We make, wait, what did? So would we reduce our acetyl-CoA levels? Yes, we would. Why would we reduce our acetyl-CoA levels?
Because we don't need
So dehydrogenase converts pyruvate to acetyl-CoA.
Yes. It would back up, right? And so we would not make them. We wouldn't want to use them if TPP is low. If TPP is low, we just wouldn't. We just wouldn't make it. It would back up as what? Pyruvate. Would back up a pyruvate. And so that's exactly what C is. So if pyruvate levels are increasing, then acetyl-CoA levels have to be decreasing.
TPP is attached to what E1, which is pyruvate.
E1. TPP is part of E1. It's the first step. Okay. All right, so those were supposed to be my easy ones, but y'all had them already. Okay, so let's go to some a little more challenging ones. Have y'all taken a look at these?
I have.
Okay, so I'm actually gonna run the iClicker because I wanna know what y'all think about these. So y'all just remind me, some of them are choose multiple, some of them are choose one. So help me remember to switch back and forth. Okay, so this is just a choose one, so it's a multiple choice. So tell me, what did you think before we go over it? What did you think the answer was? This one was sort of tricky.
All these, aren't all these correct?
Hmm?
Aren't all these correct?
There's only one correct answer to this question.
I'm saying like they're all, what I mean by that is like they're all like,
They might be. They might be. They could be. I'm not saying that they're all true, but they could be. Okay, come on, y'all. Y'all should have already read these. I'm looking at the numbers and I'm thinking maybe that didn't happen. I'll give y'all another minute. It was cold in my office. It's kind of warm in here. You never know. All right, y'all, 15 seconds. You put different answers. That's okay. That's okay. We're going to talk about it. I'm not grading this. It's okay. All right. So I want to see what we picked as the right answer. So we're kind of torn between B and C. Okay. Well, let's talk about it. So let's talk about A. You didn't like A and you didn't like D. So let's see. Which of the following best hypothesizes as to why the activity of pyruvate dehydrogenase is so tightly controlled by the cell? Some statements may be true, but you have to pick the correct one that supports the explanation. So we said it wasn't A and it wasn't D. So A says tight control of pyruvate dehydrogenase activity is needed because the enzyme is readily regulated by allosteric effectors and covalent modification.
I mean, that doesn't explain, I don't think that explains why.
This is how it's regulated, but not why. Right?
Yeah.
Okay.
That's what I was-
Okay, so D, tight control of pyruvate dehydrogenase activity is needed so that people do not develop vitamin deficiencies.
That's, no, that's good.
What are these vitamin deficiencies due to? Lack or absence of what? The coenzymes, right? All of our coenzymes. What's that? Yes, from
That's more to do with your diet than your,
diet. That is right.
you have more to do with your diet than your actual enzyme.
Correct. So it's not controlled. Okay. So you do, if you don't have these coenzymes, yes, you're going to develop deficiency diseases, right? That's how they were originally identified. That's why they have two names. Okay. So B and C, let's talk about B. Tight control of pyruvate dehydrogenase activity is needed so that pyruvate dehydrogenase can catalyze the reactions that produce acetyl-CoA. Does it do that? Does it catalyze the reactions that produce acetyl-CoA? So this is true, which enters the citrate cycle. Is that true? Yes, in order to provide energy. Is that true? So this statement is true. Okay, let's look at C. Tight control of pyruvate dehydrogenase activity is needed in order to coordinate the energy needs of the cell with production of acetyl-CoA. Now we're really getting an explanation here, right? Because this is telling us if we have a high energy in the cell, I always do that, E-N-E-R-G. I always put two E's. I don't know why. High energy in the cell. Do you want to go through citrate cycle? No. High energy means no citrate cycle. What's the best way to stop a cycle? Before it starts, right? Remove one of its precursors. If you can do that, then you can very tightly regulate the citrate cycle, right? So it's dependent upon the state the cell's in to start with. If you've got low energy, yes, let's rock and roll, right? But if you've got a high energy charge of the cell, you got lots of ATP, right? Do you need to go through citrate? The energy of the cell is all about the amount of ATP that's present, right? It is an inhibitor. It is an inhibitor. What else is going to happen? If we have high levels of ATP, what else is going to build up that you see on those diagrams in your book that we know are inhibitors of the citrate cycle? NADH is going to be an inhibitor. So why are you going to have high levels of NADH? Well, you have lots of ATP, so you're not going to be going through the electron transport chain. If you don't go through the electron transport chain, you're not going to regenerate NAD+. So you're going to end up with lots of NADH. Same result, high ATP, high NADH. Okay, all right, so what do we say? B or C now? Now we say C. So B is true. The others, D was not true, and A was not quite true. But B was true, and C was true, but C is actually the explanation. Okay. Which of the following statements about NADH and its effect on the citrate cycle are true? Choose one or more. So put your answer as A, comma, B, comma, no spaces. Okay, so always do it just like that, no spaces.
Should we also, is it outstanding affected by ATP and ATP outstanding affects pyruvate dehydrogenase?
Yes.
So it's kind of like both.
Well, it's more the calcium levels that are going to regulate the kinase activity, but ATP can regulate kinase activity.
Kinase, and then it had to reverse, like make it active again, and now it just have calcium.
Yeah, so it's really mainly calcium. It's really mainly calcium, but it will have an effect. But calcium is kind of the major regulator to reactivate. So in your muscle cells, right, when you start exercising, how do your muscles know when to contract? Calcium influx, right? So if you have a calcium influx in the cell, what you want is you want to remove the phosphate group, right, from your pyruvate decarboxylase, so that you can go through and you can start going through the citrate cycle again. But it's not directly regulated by ATP. It's really regulated by calcium. We're going to talk about, we're going to put together all the regulation at the end. Trust me, we're going to put it all together. Okay, you have less than 10 seconds. I'll give you 10 seconds, so 2:05 to go ahead and click in. We got side conversations going on. Okay. All right, so what do we say? Let's go through each one. Choose one or more. So high concentrations of NADH will lead to an increased amount of acetyl-CoA being produced due to NADH activating the pyruvate dehydrogenase enzyme? No, 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 it's not gonna, no. This is gonna be the opposite, right? Okay, so not B. NADH will bind to the enzyme citrate synthase, and when there's a significantly more NADH in the cell than NAD+, less citrate will be produced. Yes, yes.
Yeah,
That's correct, right? If you don't have NAD+, none of the citrate cycle is going to go. You need the NAD+ so that it can pick up the electrons. So the more NADH you have, the less available electron carriers you have, right? Okay, so increasing NADH concentrations will inhibit the citrate cycle. Yes. Low concentrations of NADH will result in lower concentrations of alpha ketoglutarate due to inhibition of the isocitrate dehydrogenase enzyme.
No.
No, that's
Correct. Okay, so what did, so we said B and C. B and C.
I said C and B.
B, C, and yeah, that's what I'm going to look for in B and C. There it is.
Not sure.
B and C. All right, I think I got them all. Because it recognizes uppercase and lowercase. So if you stick to the uppercase, it's a little easier.
I'm not sure. Is that
Yours came in as uppercase, I do believe. In terms of the regulation of the citrate cycle, which of the following statements is false? I'm trying to keep the chat. All right, so which of the statements, oh, I did that wrong. Multiple choice. Okay, so which of the statements is false? If y'all know of any other cheesy math jokes, please share them with me.
I know.
Because
I know
Charles
what
loved that math joke so much. I'm like, I gotta go find some more.
Why is, I know one. Why is,
Okay.
why does 6 never want to sit next to 7 at the table?
Oh, this one I know, because 7, 8, 9.
Correct.
Yes, because 7, 8, 9. I remember that from, oh my God, a long time ago.
I
That's a
remember
good
an
one.
old
That's
ass-
for
I
sure.
remember an old cartoon. They say cartoons can't teach you anything.
I had no idea math jokes would make him so happy.
I guess that's his.
That's his thing.
I guess. I mean, maybe it's a sign. Maybe it's
What did, oh wait, oh, Hannah's got one. What did the triangle say to the circle? I don't know. What did the triangle say to the circle? You're pointless. That's good. That's good. Thank you, Hannah. That's it. I think he's going to really enjoy that one. All right, y'all ready? We got 5 more seconds. That's awesome. Okay, so what did we say? Which of the statements is false? Oh, so y'all think C. C is correct.
hooray.
C is correct. All right, so let's talk about them. High concentrations of NADH and low concentrations of NAD+ in the cell inhibit the citrate cycle. We just said that, right? We just said that. Increased levels of ADP stimulate the citrate cycle.
That is awesome.
That's also true because if you have high ATP, what do? You inhibit, right? So the opposite's true, okay? All right, increased calcium ion concentration inhibits the citrate cycle. What were we just talking about?
Calcium ion is help.
Right, because Ca2+ triggers muscle contractions. Right? So if the muscle contracts, what does it do when it contracts? It uses up ATP. So if you want to replace it and you don't want to fatigue your muscles, you need to give it some more ATP. So those calcium ions are going to trigger or promote going through the citrate cycle. So that one is incorrect, which makes it correct. All right, increased concentrations of succinyl-CoA and citrate inhibit enzymes in the citrate cycle, thus inhibiting the citrate cycle. This one is true. This one is true, right? Does that make sense?
Yep.
So if you're building up products, that's a feedback inhibition, right? And that's what you want. You don't want things to pile up. Because what you want to say is, all right, I don't need it for the citrate cycle. I could go ahead and shunt it off to somewhere else, right? When my energy charge is high, now I don't need to break things down. Now I need to, guess what? I need to go build, right? Whether that's proteins or fatty acids or whatever, but your body's going to use it for other purposes. Okay. So this one is a choose one. So you really had to read this one. I hope y'all have read this one. Wait, why does it do that? Because I never know what I'm doing. All right, you want the, I guess I'll put the question, right? But you got to read the whole thing. Oh no, that was 4. I took a picture of the wrong one. Oh well. Because this is not a, you can't click in on this one. You can't click in on this one. Because it's a pick 3 things.
That's multiple short answer.
Yeah, it's short answer. Oh, stop it. All right. You know what I'm going to do? I'm going to do 1, 2, 3. 1, 2, 3, 1, 2. So if you go with 1, 1, and 1, do that so that it's easy to type in. Okay.
It can also be like A, B, and C.
Can you not do ones?
Yeah.
You can. That's what. Yeah, that's what I'm saying on the handheld.
I guess. Yeah, I can.
Okay. Give you until 2 minutes. Okay. All right, so we have our individual, right? They need to produce more functional red blood cells, right? And those blood cells need heme and they need iron. So when does this happen? No. When would your body need to make?
More oxygen.
So if you go to like a higher elevation, something like that, your body's going to start producing more red blood cells. That's one example. What if you slice your leg open and start hemorrhaging? Right? You're going to have very rapid blood loss. And so your body's going to attempt to replenish that. If you go and give blood, your body's going to work to replenish that, right? So there are a lot of different situations where that happens. All right, so we need a large quantity of red blood cells, right? So what's going to happen to, like, I just want to move it over an inch.
I think you have to minimize it.
Well, no, I need to leave it up so I can see. See, it just does that. All right. So the amount of NADH and FADH2 produced by the citrate cycle is likely to do what if we need to make red blood cells? So we're trying to go into the synthesis instead of the breakdown, right? So what's going to happen? if either the things that you intake can go through the citrate cycle to make energy, or in the case where you need it for something else, you can shunt it off. So what's going to happen to our citrate cycle if we're going to say we need some of these intermediates for something else? It's going to decrease. It's going to decrease. Well, that's a different thing. What happens? Okay, let's talk about this before we get too crazy with it. When we're going to go through glycolysis, right? Glycolysis, right? What are we going to make from glycolysis? Go this way. We're going to make pyruvate. Pyruvate goes into mitochondria, converts to acetyl-CoA. Okay, we have acetyl-CoA, comes in with oxaloacetate, and we make citrate, right? What's going to happen? At what point would we stop this cycle if we wanted to make heme? Succinyl-CoA. We're going to go all the way, and this is multiple. I'll just put multiple arrows, right? We're going to go to succinyl, CCY, succinyl-CoA, right? At succinyl-CoA, we're going to stop the citrate cycle, and we're going to take that succinyl-CoA and we're going to shuttle it away for heme synthesis, right? So the amount of NAD and FAD2 produced, right? You make another NADH here, you make an FADH2 here, right? So that it doesn't get to that point. So these are never made. It's basically like the second half of the citrate cycle doesn't happen. So those two aren't made. So your level of NADH and FADH2 produced is likely to decrease.
But maybe it's like released from the third, fourth reaction.
It is.
And also the eighth, so that's why we decrease.
Right, because you're going to make two NADH, NADH, and these will be made. Those two will be made, but these two will not. So you're basically halving the number of energy carrying molecules, right? We're not saying it's going away, but we're decreasing. Okay, so, and we're doing this because succinyl-CoA is being depleted from the cycle. So the hardest one was to answer the very first part, right? Yeah. Once you understand where it's being shuttled off, then you can backtrack and answer. Yeah? Okay. So this is, I think this is our last one. Yes. So our succinyl-CoA right here is being depleted from the cycle, from the citrate cycle, and pulled over to heme synthesis. Because remember how it's set up here? It provides metabolites for many biosynthetic pathways. One of those pathways is heme synthesis. Okay, good. All right. Let's try number 5. And I have it on short answer.
So for the first, like when it has always think that it's false because it's 8 that are being transferred, but yes, it's still true that 6 are. So I believe she's controlled. See, that's where I'm like.
Take your pick, and we're gonna talk about it. Take your pick, and we're gonna talk about it.
Oh, and so just kidding. I did.
It's okay. It's a good thing we kind of drew the citrate cycle. Now we don't have to draw it again. It's kind of nice. We sort of drew it. Is there anything we need to bring for the exam? No. So just like on every other exam, one sheet of blank printer paper or notebook paper or whatever. If you want a calculator, there's no math. There's no, I mean, you know, If it floats your boat, go ahead. It's been with you for so much, you know, it's like your talisman. You can bring it, it's cool with me. But I will, just like you got that periodic table, right? And the codon chart, those pathways will be like next on there, okay? If you print that out, that's what you're gonna have next to you. So like if you wanna practice and study, have it next to you.
Well, like I memorized it, so then when you said that, I was like, well, then what do we need to know? Well, so I was so like the innate division, you know, that's.
Well, yeah, I don't give you everything. I don't give you everything on the pathway. There are some missing bits, like the exam is Friday. The exam is this Friday, Grace, this Friday. Remember we said Friday is dead day? Friday the 13th is a dead day. This Friday, this Friday, this Friday. Because next week we're doing chapter 10. 11, thank you, chapter 11. Moodle, the exam on Moodle says the 13th. Moodle still says the 13th. I'm gonna go change it when we walk out of here. Me and Moodle, there's so many different places on Moodle. Oh, I need to get on board with Moodle. All right, so we're 2 minutes, we're past 2 minutes. All right. What are the following false? For every one acetyl-CoA molecule that enters the citrate cycle, there are 6 electrons transferred in total. How many electrons can each of these carrier molecules pick up?
Well,
This one picks up 2.
2,
This one picks up 2. This one picks up
2,
2. This one picks up
2.
2. So how many does it transfer in total?
That's 8.
8. Transfers 8. Okay. The pyruvate dehydrogenase complex requires 4 different cofactors. The names of the particular B vitamins are shown with a B followed by the number, and the coenzyme that is synthesized from the vitamin is shown in parentheses directly after the B vitamin. Is this true or false? This is true. Why might some people think that this is false? There's a 5th one, but is it a vitamin?
No, it's not.
It's not a vitamin. It's a coenzyme, but it's not a vitamin. The coenzyme lipoamide is synthesized inside of the human body, and so it's not a vitamin. A vitamin is something we must eat. It is required for pyruvate dehydrogenase activity, and it is a coenzyme, but it is not a cofactor that is a B vitamin. So pyruvate dehydrogenase makes acetyl-CoA to power the citrate cycle? Yep.
I mean, that's literally the whole. Because if that one of the case, why would they need dehydrogenase in the first?
All right, products of the citrate cycle are used to power electron transport system, which can produce approximately 10 ATP molecules. That's just from one cycle.
Yep.
Yeah. Yes, that's correct. Okay. All right, so what I want, I did have one more. I did have one more for y'all. Forgot about this. Okay, so remember when I said, I'm not gonna make you memorize these enzyme reactions. I'm not. If I were to ask you a question about it, I would give you the picture. And then you would just have to look at it and figure out kind of what's going on. But you understand this because we've covered this, right? Okay, so considering the diagram, which of the following statements is true? Let's just work through each one. If we mutate histidine 274 to an alanine, would we eliminate enzymatic activity? So histidine 274, if we come up, right, is right here, 274. So if we mutate this to an alanine, are we going to have this hydrogen here that's attached to a nitrogen? We are not.
Yep.
So what does this hydrogen do?
Hydrogen bond.
It's going to stabilize throughout the whole thing. What is it doing?
It's stabilizing the.
It's stabilizing our intermediate here, right?
and keeping our substrate inside of the pocket. So if you don't have that, you are going to abolish enzyme activity. So that one is true. Yes, 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 absolutely. Okay, aspartate would behave as an acid in this reaction mechanism. So here's our aspartate, right here. It goes from being an oxygen with a negative charge to being hydroxyl. Is that an acid or is that a base? If you're going to accept a proton, you are a.
You're accepting it, then you're
You're a base. You're a base.
You're a base.
Right? The opposite would be an acid, right? Okay, so this one is false. Oxaloacetate would be repulsed from aspartic acid in the first step of the reaction mechanism. They both have negative charges. They both have negative charges. And so if you had a negative and a negative,
They repel each other.
they would repel each other. So this one is true. Okay, that C is true. I could get that right. Okay. All right, D, the histidine residue would hydrogen bond to acetyl-CoA throughout the reaction mechanism, stabilizing the enolate intermediate. What did we just say happens in A? The reason mutating your histidine residue in A would cause a huge problem is because it's no longer going to hydrogen bond and stabilize our intermediate. So D is also correct.
So only B is correct.
Only B was incorrect. A, C, and D were all correct. Okay. So somebody in here had the question, right? Like, if I gave you all the reactions, what am I going to test you on, right? Right? So I wanted to give you a little bit of ideas on what you could do. And can I not extend this page? How does it not do that? But so things to kind of think about while you're studying, right? If you pick the cycle, so say you're going to pick citrate. When you're analyzing the citrate cycle, can you think about which reactions are energetically favorable? Which ones are irreversible, right? Those are important control mechanisms, right? So you can think about what's reversible, what's not reversible. You can think about reactions that produce our electron carriers, our reduced, right? Electron carriers. That's really important. Oh, the ones that produce CO2, right? That's going to be important. So substrate level, LEV level phosphorylation. If you're going to do substrate level phosphorylation, what are you going to be doing? In citrate, you're going to be making what? You're going to make GTP, but what if, I mean, I'm just picking citrate because that's what we're on, but it's also on glycolysis, ATP, right? So this is citrate, this is glycolysis. What, substrate level phosphorylation? Oh, so we're going to put it together. Well, what is substrate level phosphorylation? Is taking a phosphate group from your carbon containing, right, and it transfers the energy of that bond onto ATP or GTP, and then that can be spent in the cell because that's good energy currency for the cell. Another one that's really good that you can think about, whether it's the citrate or it's glycolysis, you can think about regulation. What are our positive and negative regulators, right? And does it make sense? There are some things where it's the product of the reaction, that's an inhibitor. There are some things that are downstream allosteric inhibitors. So like ATP, right? Or NADH. Okay. So like NADH will outcompete NAD+ for binding on lots of these different enzymes.
What about the,
So what can you make? Can you make a prediction about regulation and irreversible reactions? Those need to be regulated.
Yeah, you can't go this.
Well, think about it. Think about it. If you want the cell,
Irreversible regulation need to be irreversible actions need to be regulated.
irreversible reactions do need high levels of regulation. And if you think about it, what points do you want to regulate in these energy conversion cycles? You want to regulate the ones that are expending the most energy, right? You want to control those because you don't want the cycle going through unnecessary production of stuff. So you're going to have multiple regulation points by multiple different substrates, right? In order to control the pathways. So when you start to make these kind of connections, what you're going to see is tons of overlap. And that's going to help you solidify the important reactions and how they're regulated, I promise. So
What about
this is a good,
No, like the rate limiting step,
the rate limiting steps, yeah.
which is the
The third reaction
Yes.
in citrate. Yeah. Okay. So we good?
What about like coupled reactions?
Yeah, what about coupled reactions?
Like which one?
Well, yeah, you should think about that because that really only happens in what? Right, so glycolysis is the only one that has ATP investment. All the other ones are production of ATP. So yes, yes. To use ATP. So in the first part of glycolysis, you were going to use ATP. You're going to use up those two. And in the second stage, you're going to make 4 through substrate level phosphorylation. So those first 2 where you use up ATP, is that substrate level phosphorylation? No. Substrate level phosphorylation is making ATP, not consuming it. Yeah? Okay. Any questions? I think y'all did really well with these. I think I need to grade. Do I need to grade this one? Did I not grade the last one? Which one was this?
that was the question. That was the
Was it A?
yes. No, it was A. It was no, oh wait, I know.
Which one was this?
This was supposed to be. This is supposed to be. No, I think that was supposed to be A. That's number 5.
That's number 5 because we didn't do number 6 as a thing. We did number 5. So number 5 should be A. That was our incorrect answer. Okay, good. All right, that's all I got for y'all. Good luck studying. I will see you if you want to come to class. We have never had more people than want to be in class for an exam. So if you want to be in class for the exam, just come. There'll be room. Okay, just come. If you think you might have problems, please come to class and let me help you. You know, I
just decided to update right when I started.
oh my God, that's so stressful. That's so stressful. And then final exam is going to be on paper. We're on Wednesday or Thursday. I don't remember. I think it's Wednesday. I think y'all are right. I think it's Wednesday. I think it's Wednesday. All right. Okay. Anybody on Zoom? Any questions? I'm gonna go ahead and shut her down.
Dr. B. Hello?
Are we coming to class for the final? It'll be in a different room. I'm working on that. It'll be in a different room, but you will have to come to campus. I think it's gonna be in the Pelche Auditorium. I'm gonna let y'all know for sure on Moodle, but so you'll be coming to Pelche for class to take the final, if that makes sense, Taylor. Good. Okay, anybody else? Did you say there's something false in answer B? No, this is a true statement because lipoamide is not a B vitamin. It is a coenzyme, but it's not a B vitamin cofactor. So it sounds kind of tricky, but B is correct. Okay, anybody else on Zoom? Do we have to memorize glycolytics intermediates for anabolic pathways? Yes, you do need to know. You do need to know where things are being shunted out for those anabolic pathways. There's only three. There's only three different intermediates. So yeah, you do have to know where they come out to go into other pathways.
What do you mean, like for the aerobic and anaerobic pathway? Yeah, like for the, depending on whether or not they're aerobic or anaerobic.
Well, that too, but like the different metabolites in in this process, right,
Oh,
can be shunted over to be used for different things,
oh, okay.
right?
Like
So you
NADH,
do,
FADH, or
no, like we talked about succinyl-CoA today,
oh,
being shunted over for heme biosynthesis, like that.
is that like in the slides? Or is
Do we
that
have to
with the
name,
reading?
no, the serine, tyrosine, no, no, no. Okay, anybody else on Zoom? Okay. Oh, so for question 3, you say that calcium stimulates citrate. Yes, that is correct. Because it promotes the enzyme that dephosphorylates our pyruvate dehydrogenase.
Can you hear me, Dr. B?
Oh, no, I can't. I didn't know you were speaking. I'm sorry. I didn't do that. Hold on. Try again.
I kept trying to ask my questions because of
Go ahead.
Alright.
I'm sorry.
Is it also true that calcium will stimulate the citrate cycle because calcium activates insulin, and insulin is associated with increased glucose uptake. And if you have increased glucose in the blood, you're trying to make more ATP, which is through
Well,
glycolysis.
not necessarily, because just because you're pulling in glucose doesn't mean that you're going to be making ATP. You could be making fatty acids instead. So you could be using it for other purposes. It just means that it's coming into the cell. So it can, but not necessarily.
So I'm sorry, what was the reason you said it stimulates the citrate cycle again?
Because it activates pyruvate dehydrogenase, right? Because we know that when pyruvate dehydrogenase is phosphorylated, it's inactive. So there's an enzyme that goes in and phosphorylates it. So if you have high levels of calcium, you are going to stimulate the enzyme that dephosphorylates it.
Okay, thank you. That's all my questions.
Okay, cool. All right, anybody else? I'm sorry, I didn't put the sound on, Tristan. Okay, good luck. Send me an email if you have any questions. See y'all maybe on Friday.